Ata mare everybody, and kia ora koutou, ko hiu tako ingoa, he kaiwhaka a kaumatua aho, ki manaki whenua. Morning to you all and welcome to the third of our biocontrol of weeds, or weedy, it's not so much biocontrol, but weedy seminars that we have. And today we welcome in Amy Vaughan, who is a postdoc working with us here at Lincoln. Amy is host from the UK, in Newcastle, of all places, Coles to Newcastle type of place, I guess. But Amy's come over, she did her PhD at Lincoln University and then has come in to join us as a postdoc in the molecular team. And so Amy's going to talk to us, as you can see by the title, Predicting Invasiveness Using High Priority Exemplar Species. So take it away, Amy. Thank you.
Hi, so yeah, as Hugh was saying, my talk is going to be on predicting invasiveness using high priority exemplar species. So first I'll just go over the team. This work is part of a GENOMICS Aotearoa project in Invasiveness, which is co-led by Manpreet Dami at Manaki Whenua and Angela McGoffrin at Waikato University. Myself and Ellie Pavizia are both postdocs, and as well we have Tony Smith, who's our advisor in all things machine learning. So we're looking at utilizing genomic data for the prediction of species invasiveness, and this is with the aim for fast, accurate applications at the border. So today I'm just going to take you through the background of what the problem is, the current approaches, and where we can take it. So thousands of species a year come through MPI, which is why it's currently so difficult to prioritize, maintain, and keep on track of the threats to the border. This project is only recently underway. Myself and Ellie both started late in 2021, so my talk is just going to focus primarily on where our work will fit in and what biosecurity applications we're looking to achieve in the long term. So invasive species themselves, they threaten native biota and impact primary industries. The number of mammalian and insect pests in New Zealand continues to grow, and since their introduction, numbers have exploded. So as increased trade and climate change increases, this also increases the vulnerability of new border controls to exotic organisms that then can impact health, agriculture, freshwater, and indigenous flora and fauna. This isn't restricted to any biological kingdom of phyla. Plants, invertebrates, vertebrates, and fungal invaders threaten fragile ecosystems across the globe, and these pose many difficult challenges for borders to restrict the movement and introduction of these pest species. So I'm just going to quickly go over what the mechanisms of invasiveness is in all species. This is a three-stage approach that's necessary for a successful biological invasion, and this is broken down into the first, second, and third stage, which are introduction, establishment, and then spread. The introduction of an invasive species is characterized by the escape or release of an organism to a novel location, primarily as the result of human activity. This can be by travel networks, global transit through shipping or air freight, or even as a deliberate or beneficial approach to biological control. Post-introduction, a species must become established by starting a self-sustaining viable population. It's generally a really slow phase where introduced species proliferate at very low numbers for quite a long time. This lag allows the species time and opportunity to locally adapt, undergoing genomic selection, and acclimating to a new habitat. After the species has established and the lag phase is over, generally a species must disperse and expand outside of its initial invasion area and increase its range. This is generally what can cause large-scale economic costs or damage to native biota and habitats. With humans moving around the globe and this ever increasing over the last century, the circumstances for these events of a successful invasion into new territories without natural predators or barriers for incursion, this have increased, and that's allowing more species of primary concern to travel around the globe and at faster pace. So when we talk about a mechanistic ability to invade, it's about the traits acquired by the organism to prevail. Phenotypic plasticity refers to the ability of a species to increase its fitness in new environments, and these are fundamental mechanisms that rely on the suggestion that a trait gives an advantage to adaptation and invasion. This diagram on the left here gives an example of how different genomic plasticities can benefit a population in either a general species competition or invasion event. So we have on the left here variable species, with red being high levels of genome plasticity and green being low levels of genome plasticity, having stable enough populations, and then after a major environmental change, which can either be initiated by climate change or introduction of a new species to a new habitat or environment, we see that those species with increased genome plasticity have a more ecological dominance, whereas those with less plasticity have more decline. In genomic terms, we're looking really at gene duplication events, SNPs, and acquired traits that have environmental survival benefits. So a SNP is a single nucleotide polymorphism, and it represents a change to a single nucleotide in a gene. These have been implicated in pesticide resistance in some invasive plant species already, especially here in New Zealand. Gene duplications as well are also important sources of novel phenotypes and are identified as prominent features of evolution. So an increased gene copy number allows for increased plasticity and an increased opportunity for adaptation into new environments. These selection events are often associated with the lag phase that I mentioned before in establishment of an invasion, and it allows for a population to build up preferential phenotypes, be that metabolic, nutrient acquisition, or defensive traits that could pose a benefit against native host ranges. So I'm just going to talk a little bit about Aotearoa New Zealand itself and globally the impacts of invasive species. So according to a 2016 Nature Communications paper, invasive insects cost a minimum of $70 billion per year globally, while the associated health costs exceed $7 billion per year. Though this has likely changed in the 6 years since publication, and the fact that we've also had an invasive pandemic kind of happening across the globe. Another 2016 publication estimates that, excluding overseas territories, New Zealand has the second highest number of invasive alien species globally, which is only second to the United States. Looking specifically at numbers from New Zealand, MPI reported in 2021 that the economic costs of pests in New Zealand increased from 1.5 billion per year in 2009 to 4.3 billion in 2020, with a total economic cost of pests to be 9 billion in the year 2019 to 2020, which is 2.9% of GDP. So this has a really high economic burden on New Zealand. And as global warming continues to worsen and range-restricted species have more opportunity to expand into previously uninhabitable areas and be introduced to Aotearoa's unique ecosystem, we're projecting that these numbers are going to continue to worsen, and the risk is in fact escalating. Currently, ecological techniques are utilized at the border to predict these invasion pathways, but as all these climate changes happen, these standards change and allow for more chance of an incursion. So currently there are a few different prediction tools that are utilized for monitoring and identifying risk. First, there's environmental factors. Climate matching models are used to compare locations of similarly matched climates to narrow the range of predictions to where an invasive species may emerge. So an example of this would be predicting the risk relative to the spread of fall armyworm. One of the main problems with this is as temperatures are increasing, so the line between climate differences are shifting. Temperate climates are increasingly at more risk than they previously were to invaders from subtropical and tropical climates, meaning that this becomes more burdensome to use at the border. Next, we have risk pathways. So predicting biotic invasions has been based largely on identification of species that already have a record of invasiveness abroad. So therefore, identifying risk pathways is a key factor in current prediction methods. Monitoring of these risk pathways is essential in the control of pest species such as Queensland fruit fly in fruit shipments from Australia to New Zealand as an example. As well as this, we also have trace-based statistical profiling, which looks at species distribution, morphology, or even behavioral traits. This then becomes quite difficult when you're crossing taxonomic groups. So it's quite difficult to underpin one or more traits that are uniform to the success of the unwanted species once you're bridging the species gap. So this becomes quite difficult if you're looking outside of a genus. So what are we looking for in a predictive tool? We would need something that would encompass the both, something that looks at traits, risk pathways, and environmental factors, and is genomics the way forward? Recent research is starting to show that there's clear associations between genomic changes and invasiveness. So by investigating genomic approaches, we can break this down to the gene level or lower and looking at the fundamentals of an invasive species to predict its risk. Populations are established generally in the lag phase, as I talked about before, and this is where the gene selection and adaptation generally occurs, with favorable traits outcompeting and prevailing within populations. Genomic evaluation of populations already established could therefore be used to predict for those that are currently a risk or could then be a risk in the future. So MPI's risk assessment process, it focuses on first identifying a risk. Is the species present on a list of pests or diseases of concern? Is it already present in New Zealand? And if it is already present in New Zealand, isn't already present, can it establish in New Zealand? After this has been established, the potential invader moves into a risk-based assessment. What is the likelihood of entry into New Zealand? This then leads into exposure and establishment assessments. So what is the likelihood of establishment and spread? If there's seen to be a risk, an assessment of the impacts either socially, environmentally, and economically after spread is then undertaken. And the results of all of the above assessments feed into a data matrix that they then have to determine measures to implement at the border or other pre-border sort of measures to mitigate and manage these risks and the downstream effects of these measures as well. So initially, what we're proposing is we would be targeting the initial entry prediction step, so comparing genomic data from invasive populations that we already know of. As well as this, we'll then be looking at targeting potential mechanisms of impact. Is there pathogenicity on novel hosts, which are present in New Zealand? Any resistance to pesticides, which could be a problem? And what effect in the long term would this have on New Zealand? So the aim of this project is to take advantage of high throughput genomic approaches to identify signatures that might predict invasion success in a set of high priority exemplar insects. We aim to incorporate such signatures into a predictive machine learning tool to assess invasion and establishment potential. To start with, we're using fully available genome sequences from GenBank repositories and other insect repositories of known invasive and non-invasive species. And that's to develop an invasivenesses pipeline and ultimately a machine learning framework to predict the risk of a species to New Zealand and then classify this risk into high or low risk. We're modeling this initial framework from a previous study that utilized gene expansion along lineages, which is the duplication of genes in gene family, and this would infer evolution and attribute potential changes in gene families to an increased risk as an invasive species. So why machine learning? Machine learning allows software applications to become more accurate at predicting the outcomes, and it's really, really good at predicting patterns within large data sets. The characteristics that increase the risk of a species' ability to establish and spread, they're not really currently well defined at the phenotypic level, nonetheless at the genomic level. Because of this, the ability to feed information from different genomic data sources would allow for a large-scale pattern detection of mechanisms that are currently undefined and unidentified. So this overall will allow for processing and prediction scores of large data sets with relative ease and speed. The large amounts of border incursions that we have here in New Zealand that I mentioned earlier as well, these are both factors to consider, with ease and speed being really beneficial for MPI in the long run. So initially, we are using whole genome sequences to develop this pipeline with the aim to expand into different data sets to allow for a more comprehensive tool that encompasses more traits and maybe identifies more phenotypic changes. By looking at SNPs, we can move deeper from the gene level into specific base pair changes that can alter gene expression and protein production and would be an important data set to integrate. As well as SNPs and genomes, we also are hoping to integrate microbiome data sets Recent advances in study of host associations reveal that microbes are essential for exploitation of novel resources and environments, identifying trends in these biome communities, and then finding those in relation to invasive and non-invasive would aid in general predictions and utilisation of novel resources that could be impactful in New Zealand. So for this project, we have 3 carefully selected and continually intercepted species that we are going to use as biological models to start with. We've got brown marmorated stink bug here on the right, and also Queensland fruit fly and Drosophila susukii. None of these species are currently established in New Zealand, but they are all high risk and continually intercepted at the border as well. In one part of this project, Ellie, the postdoc who's based at Waikato University, will use population genomic data sets of the 3 target species I just mentioned to predict invasiveness. So generally, population genomic approaches can have broad applications in the study of invasive species. For example, such data sets can be used to trace sources of invasion. I have this map here on the left, which is some work that Ellie's recently been doing in looking at composition of brown marmorated stink bug and location of these samples as well. They can also be used to understand how evolutionary adaptation can help invasive species take over new geographic ranges. So in this project, Ellie is currently using different approaches to scan the genome of 3 target species to find polymorphisms such as SNPs in the genome that are associated with successful invasion. She's then going to be looking at whether or not these genomic SNPs are associated with certain biological functions such as pesticide resistance or higher metabolism that can potentially help with initial establishment after the spread of an invasive species. By generating this information, we're kind of then aiming to train the machine learning framework to predict invasion potential in other species. So one of the main intended outputs of this pipeline is a predictive score of invasiveness. Such a score could be applied to species that are imminent threats because of a new trade pathway, or a species that might have range expanded recently, but not much is known about whether it would be a problem in New Zealand yet. As well, this would be really useful for species that are in New Zealand already but may become a problem in the future as New Zealand's climate becomes warmer and more hospitable. Based on discussions we've had previously with MPI, this would target on the first step in the risk pathway assessment, which is likelihood of entry. We're hoping that the information can help develop better management or biosecurity outcomes with potential for any mechanistic insights. For example, we might be able to identify characters of the species that make it more invasive, such as metabolic extension or gene families involved in sensory function, or potentially even new pathogenicity islands. The tool that we're proposing as well would be particularly useful for species where we know the least, such as fungi or even insect species that are not global pests yet, but are starting to expand their host range. That's really where we're hoping that this would be the most useful. And while we're currently working with the 3 insect species that I mentioned before, ultimately the aim would be that the tool would end up being species agnostic, meaning that it could be applied to all organisms. Yeah, so thanks. I just want to acknowledge MPI and the advisory group and thank everyone who's been involved so far.
Excellent, Amy. Thank you very much for that. That was very interesting stuff. I'm staggered by the figures that you presented, at the beginning about the costs, the numbers of invasions and the costs in New Zealand. They're pretty scary figures. I've been quoting figures for years about the cost of weeds to the New Zealand economy and things, and you've actually come up with figures about just general invasive species, which is just truly amazing. There has been one question from Fiona Hennessy. Now, I've got a funny feeling you might have actually already answered this one with the last bit of your talk, but she's saying you refer to insect species, but does the process work for invasive plant species and other organisms as well?
Yeah, so that's the general aim, is that it would be able to go into other species, whether it be plants, fungi, higher eukaryotes. That would be the general idea, and we're hoping that because we're looking not just at the gene level and we're looking at these polymorphisms that maybe it's a bit more transferable between genuses as well. So that would be the general aim, would to make it species agnostic.
Cool. I also wondered about, have we, I don't know whether we've used it in our team or whether there's a plan to use this kind of technology for deliberately introduced exotic species, which we do in terms of the biocontrol of weeds. Do you know much, are there plans to do that sort of stuff, or have you done that kind of work before?
I haven't done that kind of work before, but we've definitely talked about it because it would be a very cool sort of model system to look at, because you don't quite often get to have a look at a controlled invasion. So those are definitely conversations that we've had, and it would be something that could be quite useful for it.
Yeah, I must admit it'd be really interesting to actually see what it's like for a controlled invasion where we actually deliberately release things sort of almost all at once across a wide part of the multi. So yeah, it would be very interesting to see what the predictions would be using this modelling system. Okay, looks like that was the only question. I don't see. Oh no, we have some more that have appeared. Oh, saved my bacon. I was thinking, what am I going to do now? So David Hira, who I think might be with MPI, but David has asked. Oh, what happened there? No. Oh, there it is. Sorry. What depth of genome data is necessary to feed into the analysis? Is genotyping the sequencing by sequencing? Sorry, is genotyping by sequencing also an option, or does it have to be a whole genome analysis?
No, so GBS is definitely something we've talked about. The idea is that we can have all of these other data sets as well. We've already talked to MPI about what sort of data they tend to generate as well. The reason that we're starting with whole genome sequencing is because that's quite often what's available in the repositories, and of the ones that we can classify as definitively invasive and non-invasive, those are the data sets that we can get a hold of first off. So that's why we initially started with the whole genomes, but the idea would be to be able to transcend into other data sets such as GBS and SNPs and microbiome.
Excellent. Okay, thank you. Anna Hooper has asked, how long do you think it'll take to develop a species agnostic tool?
How long is a piece of string? Hopefully, we're going to take it one step at a time and start with insects at first, but hopefully we can then be able to train data sets. But not 100% certain how long it'll take to be species agnostic yet.
How long is your contract for? Is it like 20 years?
If I ask that.
Sorry.
Two.
Right. Holly Cox has also asked a question. What resource is needed to get a genome of a species available to do this work? I would imagine that we would need to be aware of at least the potential invasiveness of a species to upload a genome.
Not necessarily. So there's what the data set that I'm currently working with. I have 37 invasive species, 6 non-invasive species, and 99 unclassified. So with insects, I think it's with most organisms as well, you can do all your genomic data, but there doesn't have to, there might not have been the extensive range sort of assessments needed to declare whether or not it's invasive or not. I imagine you need quite a lot of data to be able to say definitively this is an invasive species or not an invasive species.
Okay, thank you. Manpreet's asked a question, so I think the boss has asked you a question. Has there been other research in this area where predicting an organism's ecological characteristics based on its genetics have been successful?
So based on its genetics, there is a paper that I mentioned that we were basing the initial stages of our study on. They use whole genome sequencing to predict invasiveness, and I think that was in 2020, and they found that if they were looking at gene expansion and contraction events, which is the duplications, that they could find genes associated with defense and metabolism that were associated with invasiveness. Their data set was quite small, and we're hoping to get more data to feed into it and have more data set types that we can feed into it as well. But there is sort of a basis that this is doable and has been looked into previously.
Excellent. This question may take a bit for you to answer. So Rosie Mertz has asked, could you go into more detail about SNPs and its use? I don't know how long it's likely to take to do that. If you feel it's going to take a while, then feel free to answer, Rosie, after the webinar.
So SNPs, I'll just do a brief coverage, cool. And then if I can send an email around afterwards, that's okay. So SNPs kind of what they are is just you have a gene and then one single change in a nucleotide, say it would be from an A to a G or a C, that can change the amino acid composition, that can then change the protein that's expressed or produced by that gene itself. So there's an example, I think it's with, there's a certain plant that has a SNP in a gene. I can't, off the top of my head, remember the name of this gene, but it does actually, there's one single nucleotide change means that this plant is now pesticide resistant to a specific pesticide we have here in New Zealand. So all it takes is one single nucleotide base to change an entire gene that can then have implications downstream. So while looking at a whole gene is very important, whether or not it's there, whether or not these changes are happening, we're finding that on a deeper level that even the general consensus, the makeup of these genes are quite important as well. Yeah, I think I could probably go into more detail afterwards because it's quite a lot of explaining. But generally, it's just because you have these very, very small changes can then lead to very big consequences downstream. So they're very applicable for when you're looking at trait changes and phenotype changes in populations.
Cool. If Rosie wants to know more, she can contact you directly and for more information, should she wish. Jez Morris has also asked, one imagines that some species will have invasive traits but not be fully invasive, the fundamental versus realized niche. When you're looking for genes that correspond to invasive traits, what range of species are you looking at? How much do you look beyond known pests or weeds and look at cosmopolitan species generally?
Yeah, so we're starting by looking at invasive and non-invasive first, just so we can categorize those traits. But yeah, that's true that there will be traits that are in species that are not characterized as invasive, but that's part of the basis for the study, is that things are changing. Just because something is currently not an invasive or is declared as a non-invasive doesn't mean it doesn't have potential traits or ability to adapt to new host ranges as things are changing. So it's more of a prediction of the risk than definitively saying this is currently an invasive, but what could be in the long run? I think that answers that question.
Cool. I think that's it, Amy. I don't see any more questions coming through at the moment. So thank you very much. That was truly fascinating stuff and well done. So I think it's work that'll be very interesting to see the results of and see how you get on over the next few years as part of your postdoc. But thank you very much, and thank you to everyone for attending. We will probably see some of you again tomorrow for our final day of webinars on the Biosecurity Bonanza. Thanks, Amy.
Thank you.